Yeah, I guess.
All right, we are recording now. Good morning. How's the week going?
I was woken up by a fire alarm.
I caught some of that, so yeah. Do you live in apartments or
Fifties
dorms?
apartments.
Yeah, that's that trick never ceases. It just always continues year after year, generation after generation. I was at an ESA meeting one time and in the student hotel, someone did that. And it seems like every year they do it too. And then you get middle of the night, and one of the other students when I was a PhD student was rooming with, he like gets up to bolt out, and he's like, "Where are you going?" Or like, "Why are you not going?" I'm like, they do this all the time in the dorms. There's nothing out there. All of you are going to be standing out there at 3 o'clock in the morning. And it's like, yeah, there's no fire. Anyway, so always continues. All right. So what we're going to do today, a couple of different things. First, I just want to recap what we talked about with integument systems. Um, we will get into the molting part of that. The lecture I have for that somewhere in the IT migration, it's in the black hole space, so I will find it and extract it, and we'll come back to it. So we'll move on to sensory systems just to keep ourselves um, in check here. And then I want to spend a few minutes talking about the concept note that's due at the end of the semester. I know a lot of you probably are aware of what that looks like, but for those of you that are not, I just want to break down the 6 different criteria that I'm looking for with that concept note and what each of them means. It'll help with the distance students as well. So, to kick it off, with the integumentary system, we covered quite a bit in a short period of time. And I just want to go over some high level points of what to think about when one for the exam, but also just kind of that conversational knowledge about the physiology of the integument and how it works or how it's produced. So, we started off basically describing the primary functions of an insect egg as a structure. What are those functions? Nutrients, right? So you have those egg proteins, those vitellogen, vitellophages, right? What else?
Make sure the insect in its young stage doesn't dry out.
Yeah, so desiccation is a big problem, right? So I want to protect it from the environment. What else?
Embryo development.
Yeah, so it's protecting yeah. So there's different morphogens that are going on,
Yeah.
different gene expression, protein expression patterns, right? And then all it's a concerted effort, right, to be able to produce that developing embryo, right?
Does it also, they also have like membranous aspects of them that would allow like oxygen to get in?
Yeah, absolutely. So they do have pores, so their oxygen and CO2 has to be able to move in and out. Anything else I'm missing?
Enough to get lays.
Yeah, it has to make its way through an ovipositor, right? That's a good point, Dean.
Especially like the monoid wasps. Pretty crazy, right?
Right, yeah. Yeah, and they come in all shapes and sizes, all different colors, right? So, but those capture the main points of the structure and the function. Don't be deceived by that it's a simple structure because it's not. It's as I showed you in the illustrations, if you try to dissect eggs out or look at different X-ray imaging of eggs, you find that they're highly complex structures, a lot of different features. So we also talked about embryonic pattern formation and these different morphogens that are involved in that. So they're all maternally derived morphogens that they produce these gradients that establish the anterior and posterior regions, right? So it starts with the broad picture, if you will, the broad strokes of that oocyte. How do these gradients work? How do these morphogens work? They do, right? So Remember, we talked about 4 primary messenger RNAs and their expression for the anterior and the posterior, right? So on the anterior, you have bicoid, hunchback, right? And so those are highly expressed for the anterior. And on the other side, you have Nanus and caudal, right? So Nanus and caudal on this side, right? So both, yeah, hunchback bicoid for the anterior, Nanus caudal on the posterior. Those are highly concentrated messenger RNAs, and then their gradients decrease as you move to the opposite pole ends, right? So they activate and they repress these other segment polarity genes downstream to kind of start defining those broad characteristics. So we talked about there's this hierarchy or hierarchical cascade of segmentation genes, and they start with those maternal effect genes, again activating and repressing what we define as the broad regions of the insect. And then you move into the gap genes and the pararule genes. And then we started talking about from those, the different parasegments are formed. And then we go into the pararule genes and then the homeotic genes until you start having specifically identified characters on each body segment, right? So what happens if these homeotic genes are mutated? Samuel, you know this. It looks weird, yeah. Mm-hmm. But they also, the different expression patterns and different mutations give rise to the different features, right? So why different insects have different eggs, different species have different wings, different antennae, so on and so forth. So those expression patterns and those mutations, we can also create those mutations, right? To be able to look at embryonic development and how and where each of these structures are produced. We also talked about the difference between holoblastic and meriblastic cleavage. Remember that the holoblastic cleavage is where the daughter cells or daughter nuclei divide altogether, whereas the meriblastic cleavage is where they're still intact, right? And so insects tend to have more of the meriblastic cleavage in the embryonic development stages. We also talked about energids and how those produce pole cells and vitellogenins. We talked a little bit about gastrulation and basically how the developing embryo starts to basically collapse or twist on itself to start giving closure to the different portions of the body, but also at the same time starting to differentiate where organs and tissues are going to develop. How does the tracheal system develop?
And then branches out.
Yeah.
Branching.
It's like a plant. It's like a tree, right? So, and it works with these 2 different genes and their receptors known as branchless and breathless, right? So they work together. And just think of it as a tree growing. I mean, you got a seedling and then it just evolves into this massive network of the tracheal system producing the tracheal trunks, the tracheoles, air sacs and whatnot. We also went through briefly about the different instars and the stadia and the ferrate stages. What do each one of those mean? When we're talking about apolysis and ecdysis as we progress from egg through the instar, pupal, and adult stages. What's the difference between an instar and a stadium?
Yeah.
So with apolysis, remember that you're shedding the cuticle, the old cuticle. So that's the difference between the instar stages, right?
The stadiums or the instar is separated, or the organism is separated from the cuticle.
Right. So that's ecdysis, right? So perfect. Yeah. So ecdysis is separating from the cuticle, and apolysis is basically detaching to the next stage from that cuticle. You're on track there, Venus. Yeah, you're getting there with it. We also talked about the structure of the integument. And that's there's three main components. Talked about the basement membrane starts the formation followed by the epidermis, and then you have the cuticle on top, right? Which is would you consider to be the only living level of that cuticle? Epidermis, yeah. Why?
That's where all.
It's packed with all those cells, right? So the dermal glands and the enocytes and everything. So it has all of the functional cells, living cells, to be able to produce the integument structure. So let's see. We also talked about we finished up with chitin. So what are the three main components of chitin?
Water.
Water is part of it, yeah. Or I should say the cuticle.
Oh.
So chitin is one. Water is in there, yeah. Because it makes up whether it's a flexible cuticle or a hardened cuticle, right? So it's the water composition. What else? Proteins, right? So you got a lot of protein synthesis going on in the development of the cuticle. What else? What protects it? What waterproofs it? Yeah, wax. So lipids, wax, layer. So what's the chemical structure of chitin? What is it? You had to explain it to a non-entomologist.
It's like a sugar with
Yeah.
my group.
Mm-hmm.
I mean,
Yeah, it's a glucosamine, right? It's a polysaccharide chain, right? And it's beta linked, okay? And there's different formations of it, right? It tends to be more of a bidirectional flow for insects, but the way that they're linked or the way that the patterns are give the different cuticular structures. So yeah, it's a polysaccharide sugar, right? How is it synthesized? Trehalose, do you remember trehalose in the hemolymph? And then there's this cascade of different enzymatic reactions that convert it to chitin, right? And 20-hydroxyecdysone is involved in that. There's another key enzyme that's involved in that where insecticide toxicologists and chemists have targeted for insect growth regulator compounds. Chitin synthesis or chitin synthase, yeah. So that's the key enzyme in that whole process. You can go through all of those other biochemical steps, but without chitin synthase, the insect will never produce chitin. Okay. I think that kind of catches us up with what we talked about. We already talked about water content, and more water content produces a more flexible cuticle, whereas the more hardened cuticles have less water content. Yeah, it can be. Yeah, it depends on those hydroxyl groups, right? And the availability of hydroxyl groups to be able to do that. So, which come from water. Good work. All right. So we'll move on to sensory systems. When we talk about sensory systems, you'll see that it's mostly focusing on different structural forms of sensilla, and then how these respond to different stimuli. We'll mostly focus on olfactory and gustatory stimuli, but we'll catch up with some other types of modified sensilla through this lecture as well. If I can get it started. So again, why I start with the nervous system is because it controls everything, right? So we have discussed about the origins of the nervous system, its general function, how it works with the endocrine system, how it works with the integumentary system. And now we move into sensory systems here. But this nervous system, it has to be able to receive information across the integument to be able to work properly. And this is where the sensilla come into play. So there are receptor surfaces, small pores. They're not uniformly distributed over the body, but they tend to be these primary sense cells that are associated with a specific neuron to be able to drive whatever behavioral response that the stimuli is eliciting. So you'll tend to find within the integument here, you'll have these sense cells. They can take different forms. Of course, it really depends on what their overall function is going to be. And then again, they are connected to those neurons to be able to drive behaviors. So the sensillum is the basic unit of sensory reception. You can have sensory neurons. It tends to have the packaging or the structure has a ptormogen cell, a tricogen cell, and a thecogen cell. Right here is where I'm going. So with this illustration, again, just a general illustration, but there'll be different forms of this depending on what the stimuli that it's picking up, whether it's olfactory, gustatory, whether it's vibrational cues, infrared cues, or humidity, temperature. There are several different shapes, but the overall structure remains relatively the same. In this basic sensillum here, you have these dendrites, again, that are connecting to the axons and driving the central nervous system. For this, in this case here, it's more of a mechanoreceptor receptor, and it's picking up any type of wind movement, vibrations, and whatnot. But then you have these ptormogen cells and thecogen cells, tricogen cells here that basically encase those axons. And again, you have different structures here depending on what the stimuli is going to be. So with your tricoid and basonic sensilla, you tend to have more of the gustatory and olfactory cues. With campaniform and placoid, these are mostly on body movement and orientation. Campaniform can also be for vibrational cues or substrate movements. And then cilionic here, which tend to be more related to temperature and humidity stimuli. But overall, these sensory neurons, they have dendrites that extend to the cuticle and axons, and then again extending to the central nervous system. And again, they're structurally characterized by the external morphology as well as their functionality to a stimulus. So there are two types of chemoreceptors before we get into the mechanoreceptors. There are gustatory receptors and olfactory receptors, and they both work in a similar fashion. So gustatory receptors, these are the taste receptors. They're concentrated in the mouth parts, the legs, ovipositor. And they have neurons that are specialized for different substrates or different media, so whether it's sugar, water, or salts. And they're all activated by these G protein coupled receptors, or these secondary messenger systems that we talked about back in the neurophysiology nervous system section. So these taste receptors, they have these chemoreceptor axons that connect to interneurons and ganglia in different segments, whether it's on the mouth parts, legs, or ovipositor. And then neurons can express multiple taste receptors, okay? And next, you have the olfactory chemoreceptors, and these tend to be, order receptors, but they're on the antennae or the palps. It's kind of older knowledge of the antennae and palps. They tend to be all over the body, okay? There's some really interesting work that a few years ago out of Yale University, where they went through and they used single cell recording. We'll talk about this here in a moment. But they characterized all of the sensilla on the body of Drosophila and what their functions were, their olfactory functions were. Enormous project, right? And then they went on further to take this Drosophila model, knock out its olfactory system, and then to characterize all of the olfactory receptors and gustatory receptors of malaria mosquitoes. They knocked in those individual receptors from the mosquitoes into the fly and did the same work, all right? So really interesting outcomes from that, and how finding out that it's not a really a one size fits all, and that you can have gustatory and olfactory receptors, chemoreceptors all over the body of insects. And so again, we talked about the three different parts of the insect brain, the protocerebrum, the deuterocerebrum, and tritocerebrum. With olfactory receptors, the axons tend to terminate in the deuterocerebrum. There can be over 1,000 olfactory receptor neurons, and each one of those neurons is expressed for one specific odor. Okay. I should be careful with this because when we start talking about specialist and generalist stimuli, you can get single compound responses, olfactory and gustatory responses, or you can get blends. And this will become more important when we start talking about behaviors and pheromone blends. So a lot of your chemical ecologists will say that it is all about the blend and not just the individual components of it. So for example, when we were working through guideposts for our attractive targeted sugar baits, we went into floral blends that were in the literature already known to attract mosquitoes. And we took those blends and we dissected out each of the single compounds and then put them back together. And what you find out is you take them apart. So together, they're highly attractive to mosquitoes. You take them apart and they start having repellent activities as single compounds. Put them back together, you get that attractive property. So it's all about the blend. It's about the composition and the concentration to be able to stimulate these receptors into behavioral responses. So as I mentioned before, with these sensory systems, they tend to be highly connected to the brain. We talked about the deuterocerebrum associated with the olfactory receptors, olfactory receptors, and then the tritocerebrum is more with the gustatory receptors. And those gustatory receptors, again, being on the mouth parts, olfactory on the antennae. But again, they're all connected to the brain here and then connected to the central nervous system for eliciting those different behaviors, depending on what the stimuli is, of course. So how do these work? So what happens here is you have a stimulus molecule, whether it's for taste or for smell, and these stimulus molecules will pass through a pore in the cuticle. So say antennae, for example, can be have one pore or they can be multiporous. Okay, they tend to be more multiporous and to be able to deliver these stimulus molecules. So you have the molecule that passes through there. It's in the syncytium lymph. And what it does is it binds to an odorant binding protein. That odorant binding protein travels it or takes it to a specific olfactory receptor. And then from there, you get an action potential or depolarization event leading to that behavior from the nervous system. So just a very general kind of delivery system here, if you will. Keep in mind that most of these olfactory binding proteins, olfactory receptors, they're very specific for the molecule that's entering in that syncytial lymph. And then you also have these odorant degrading esterases to be able to break it down, remove that molecule from the odorant binding protein and stop that whole activity or action in the system. So what's happening here, and I'll talk a little bit more about where this comes into place with these receptor potentials and secondary messenger systems. But this signal transduction is basically transferring environmental energy into electrical energy in the nervous system. So again, you can get these sensory molecules to either absorb through the cuticle or to diffuse through these different pores. Right. And again, what's happening here is it's traveling through the syncytial lymph and it's binding to that odorant binding protein. The odorant binding protein takes it to a specific receptor. You get a receptor potential from that. which is driven by these secondary messenger systems, and we'll talk about trip channels and calcium here in a moment. But again, it's basically causing this membrane conductance and the depolarization event to elicit an action potential. And then there has to be an activation phase here, and that's what's happening with those odorant degrading esterases. They're coming in and dissociating that molecule from the protein. Any questions so far? Yeah, North.
One. Before we like attach to binding, how specific are those binding proteins the odorants attached to?
Very specific. So it's think of it as this is one odorant binding protein per molecule, right? And so it has to be able to differentiate what that molecule, what the functional groups on that molecule are, and then it takes it specifically to an olfactory receptor, right? So if it has to be able to stimulate the right receptor for the right response. Does that make sense? And to be able to go through, this is one of the limitations, is to be able to go through and dissect out the neural circuitry to know which of the olfactory neurons are connected to that olfactory receptor, and then what the stimulus is or the molecule to be able to drive that response. So it's not a one size fits all. Most of them are specialists. There are some generalists. So when I say generalists, we'll talk about here in a moment, is that when you get chemical blends, right, you're going to have several different molecules within that blend, right? And those individual molecules are going to associate with specific odorant binding proteins. And those binding proteins are going to take those molecules to those receptors. And when they, let's say, connect and light them up, then you're going to get a behavioral response. But they all have to work together, right? Again, it can be specialists. Most of them are specialists, but then in the blend phase of it, they rely, certain behaviors rely on, say, 5, 6, 7 different olfactory receptors or gustatory receptors to be triggered at the same time. Does that make sense? What?
I'm trying to compare this with the tubular scintillum recordings, but
Mhm.
is the odorant binding proteins, because we know from singular scintilla recordings, the SSR, that different scintilla react differently to certain chemicals. So is the odorant binding proteins for the different scintilla, are they unique to those scintilla? And that's why we see those different responses, or?
Yes and no.
Okay.
All right. Again, not a one size fits all here. So we'll talk about this here in a moment. But if you go to if you remember the EAG setup, right? So you're connecting the entire antennae, right? With electrodes, and you're recording the activity of that antenna when you add a stimulus. We don't know what olfactory receptor or what olfactory neuron is being triggered, right? So I always say EAG work is like hitting the olfactory system with a sledgehammer, right? So you're lighting everything up, right? Or it appears to be. And then with the single cell to single scintilla recordings, you can target individual olfactory neurons, but that's not necessarily giving you the information of the olfactory receptors that are associated with it, or the odorant binding proteins that are, you see what I'm saying?
Okay. Yeah, I was just curious, because if those are so specific, and so are the odorant binding proteins, I didn't know if they were linked at all.
Yeah, I mean, there's more and more information coming out in the
Yeah.
literature where people are doing the neural circuitry dissections, right? To hone in on that. I mean, for example, there's a reason why DEET is the gold standard for repellency, because it not only works as a specialist, it works as a generalist type of chemical. It has all kinds of different effects. And that's where the debates come into play of, does it affect the olfactory system? Does it affect the gustatory system? And what are those olfactory or gustatory receptors that it's targeting? It's just, that's the way it is. A lot of your compounds that we use for like synergists, right?
Mhm.
So while, say, if you want to suppress the detoxification proteins, P450. You can do that with PBO, right? With piperonal butoxide, which is a natural product terpenoid, but it also has other effects too. And so that's kind of where we get in the weeds with these things. But it's a really good question. Both of those techniques have their purpose. There's just limitations when we get into them. So you have to couple that with other behavioral studies, and that's where the molecular biology comes into play too, where you have to knock out those things. So that's where I was going with. I don't know if you were in here when I was talking about the Yale work with John Carlson's work, where he went in and did the SSR on individual syncytium all across the Drosophila body, and then he did it on mosquitoes. And yeah, so it's really impressive work. Tedious, but fills a lot of knowledge gaps. Okay, so I want to move on from this environmental energy to electrical energy. And I mentioned these G protein coupled receptors. So these stimulus molecules, they'll bind to these GPCRs, and what they do is they activate this phospholipase C here. So here's the whole G protein alpha subunit that molecule is associating with. Once the PLC is activated, it produces inositol triphosphate, or IP3 here. And what that does is it stimulates the endoplasmic reticulum to produce calcium. And we talked about this in the neurophysiology section, that calcium stimulates acetylcholine to be released. Acetylcholine is released for acetylcholine receptors, and then it starts that whole depolarization process. So you get intracellular calcium opens. What they do is they open these transient receptor proteins called TRP channels. And the TRP channels then cause membrane depolarization by allowing sodium channels to open. And then this is where the odorant degrading esterases come in. They break down this association. And then once you have that action potential, you go to the repolarization phase with potassium channels opening and bringing that membrane potential down to its resting state. Okay. So again, these specific molecules from the environment are carried to their specific receptors. Once they hit those receptors, it starts this secondary cascade to cause membrane depolarization, leading to the intended behavior from the stimulus. So as we've been talking about, you have these specialist and generalist receptors, right? And so the brain can receive information again in these two different ways. The specialist receptor stimulates the brain to know so that the particular compound is present, and just that compound alone can cause stimulation of a behavior. Okay, so this is kind of the memory phase of the olfactory and gustatory receptor system. So it's the already memory knowledge phase of the insect. And so you have a population of these olfactory neurons. They'll innervate a single glomerulus of the brain, and then the message is then relayed to higher order brain centers to process for behavioral activity. So you can have these labeled line patterns, and that is the one stimulus, one receptor, one response type of pattern, or you can have a combination, right? So where you have, again, specialist receptors, but they have to be activated at the same time to create a different response. Okay, so you have these combinations as well. And then they build off of this. So just real quick here is that it's difficult for the insect to process or accommodate every possible stimulus. And so it has to work together with these more of this combination line pattern or other receptors. And so you'll have specialist variants, and they require blends or pheromones again for behavioral activation. So as I mentioned before, a lot of your chemical ecologists, chemical biologists will tell you that it's all about that blend and not really this individual compounds. Okay, everything occurs in blends. And then you have, other receptors that have broader response to these stimuli. And these are your generalist receptors. And so rather than just binding a few specific receptors, they activate more generalist type of responses or a greater range or lesser degree of responses. So a large number of different stimuli at the same time will create these action fiber line patterns where it requires multiple receptors being activated at the same time to be able to elicit one response. So when I say generalist receptor here, it's usually not one molecule. Each one of those receptors are taking a blend of molecules to be able to facilitate that behavioral response. So this is where it starts to get complex in dissecting out what is the overall olfactory gustatory stimuli stimulus that's actually eliciting that behavioral response. And like we were talking about, Gwen, you can use EAG or SSR, but you have to couple that with other downstream behavioral and molecular approaches to be able to identify exactly what it is that's being affected. So we talked about in previous lectures, EAG. This was on your exam. So olfactory receptors on the whole, basically it measures nerve responses on the olfactory receptor of the whole antenna when a stimulus is delivered to it. So just the basic setup here again, you have two recording electrodes that you place the antenna across. There's usually a conducting gel or conductance gel that's associated with that. You have a stimulus molecule that you apply to filter paper, and you basically deliver that as sort of a puff of air across the antennae. That signal is amplified, and then you have a recording device to actually measure that electrical activity that you can analyze later on. But anyway, what's going on here is the same thing that I showed you in the previous slides, is that it's a stimulus that's binding to a protein, binding to a receptor, and causing membrane depolarization. And that's what you're measuring here with the recording device. So what Gwen was talking about, single cell recording or single syncytium recording, you can do this with both olfactory and gustatory receptors. You can do the EAG with them too. So you can replace that antenna with legs as well, or mouth parts, or proboscis, for example, you can do the same thing with. So the difference here is that tends to be a little bit more specific in that you're using an electrode to basically impale an olfactory neuron, and then you're firing that stimulus across the syncytium, and then you're recording that membrane depolarization event. Okay, works the same way. It's just a specific, more specific technique. Again, it's not telling you really what the binding protein is or what the receptor is. It's just telling you what neuron's being activated, and then you would have to use other approaches to be able to further describe what those binding proteins and receptors are in this case. So I think we went through the strengths and the weaknesses here. So what's the strength of EAG? For those of you that have completed the exam.
I mean, you can see the output of what a single factory nerve would.
Yeah, it's just a broad effect, right? It's the broad activity of whatever stimulus you fire at it, right? Whether it's single compounds or blends of compounds, right? So it's a very high throughput system to be able to screen antennal activity, okay? But even just a puff of air stimulates antennal activity. So you got to get a response that's greater than the baseline, right? What are some weaknesses?
It's not the whole system, and it could be like further interactions happening in the entire insect.
That's a good point, Honor. I'm glad you brought that up because we haven't even talked about behavioral assays yet, right? So I think I've shown you like in the Kajiput terpene study, we use that olfactory kind of tube system for behavioral. It allows you to monitor attraction. Yeah, attraction or repellency, right? But that behavioral doesn't necessarily mean it's driven by the antennae, right?
Yeah.
And so it could be gustatory receptors. It could be a variety of different things, right? Right. Yeah, exactly, exactly. So you're really limited here to just the antennae or the tarsi. I mean, you can do the tarsi as well, but that doesn't necessarily mean your behavioral response is an olfactory response and whatnot. So yeah, there are quite a bit of limitations. It's still highly used in repellency attraction work. I mean, it's a great tool, but again, there are limitations with it. Eli.
Like the Nuda cerebrum and Pita cerebrum are right next to each other,
Mhm.
and they are responsible for olfactory and gustatory
Right.
respectively. Is there any like activity that would be like an AND gate where you have like a certain olfactory signal and a certain gustatory signal? If both of those are present, there's a specific stimulus or a like specific response to that, or
That's a good point,
Okay.
Glenn.
It's possible for like plant feeding, so they may use it to find
yeah,
the plants, and then when they land on it, they can use those gustatory signals to like,
Right.
it's like confirmation.
taste basically to see if it is fitting their nutritious needs.
Right.
Oh, it's like switching between signals, like
Mhm.
they switch from like CO2 to like heat as they go.
Yeah. So all of you are tapping into, yeah, this is a perfect discussion for this because think about and this is a lot of insects. But in the beginning, I showed you that whole odor plume cascade for like mosquitoes, right? So not only like Gwen was saying, I mean, with her project, that they're attracted by some chemical cue from a plant to drive nectar feeding, right? And so then olfaction and gustation are working together, right? Even with oviposition. So you have oviposition sites, you have microbes that are producing volatiles from the water that signal, all right, this might be an attractive oviposition site, but then that's why you have these gustatory receptors or sensilla on the ovipositor, the abdomen and the feet too, because that female mosquito is like, all right, is this a suitable habitat for me to lay eggs in, right? So they're all working together with that. Even like our ATSB work that I mentioned, we can attract mosquitoes to that bait station, but you want to keep them there. You want to keep them interested because there's a short time period when they're okay, I'm attracted to possibly a food source, but they're only going to probe around, dance around on that substrate for a certain period of time, and then they're moving off, right? So that's why we add feeding stimulants, and other compounds into that bait station because we want them to rest and feed for a longer period of time, right? And I'm going to add this for our next Think and React article. This is work we did a little while back where we were trying to find repellents, bee-specific repellents, if you will, to put into pesticide formulations that would keep bees from visiting sprayed crops for a short period of time. Okay. So what we did here, again, a lot of these compounds that I talk about, these natural product compounds are going to be terpenoids, but they can induce feeding deterrence and then also antennal responses. So we did a lot of behavioral studies with this to show that a variety of different compounds from DEET to citronellin to geraniol and nerol, basically how each one of these decrease the time in a feeding zone. And then we correlated that to antennal responses using EAG experimentation, right? And showing these different stimuli. we have another follow-up study where we actually did the field work, and you can actually drive bees for a short period of time away from different cropping systems. We used melon flowers and then also knapweed in that case. But yeah, so you can do this. You can use these different techniques or to be able to kind of dial in on olfactory gustatory responses and be able to use them for, I talk about pest management, but also for pollinator protection as well. Right? Any questions? Right.
So is this like a topical insecticide that would be for something that lives on the plant?
So this was sprayed on paint, so it was contact. What is it? Was contact? It was olfactory. Okay, so they wouldn't go in, they wouldn't touch the plant or land on the plants. But insecticide pesticide formulations, they'll have their active ingredient toxicant in there, and then there's a variety of other things to basically protect the integrity of that toxicant for crop use, right? For pest management. But the idea here is that if you can add into the formulation a repellent that's short-lived, when you spray cropping systems, that you can keep pollinators from visiting them for a shorter period of time without, you only want to manipulate that behavior for a short time. You don't want to kill them or fumigate them, right? So,
But like would a like topical insecticide like you're talking about be like if you're adding a repellent, it would probably be more effective against something that is not able to get away?
I don't know if I understand where you're going.
Specific.
So yeah, so this is actually that's a good point. So geraniol, nerol, citral, isoamyl acetate. So these all make up what we know as Nasodov pheromone in bees. And so it's more of that's an aggregation pheromone. But this was a case where if you separate each one of those single compounds from that, you get repellent effects. You put them back together, and then you get more attractive effects or aggregation effects. And it's interesting, you can take and manipulate the composition and the concentration of these, where you can get bees attack each other, which was interesting because of, I mean, their social nature, you wouldn't think that, but they will do that if you don't have the composition right. I guess maybe to follow up with your question. So are you familiar with pyrethroid insecticides? So pyrethroids are derived from chrysanthemums. They target sodium channels, right? But you have these volatile pyrethroids like transfluthrin, which is used in spatial emanators, right? So they're produced, and the volatiles will repel mosquitoes, but contact-wise, they'll kill mosquitoes too because they target sodium channels, right? So in the vapor phase and contact phase. So you can have compounds that are specifically designed for a mode of action on, say, sodium channel, but they have a different action, olfactory action, where they can repel insects as well. So does that kind of get to where you were going? I'm having difficulty.
Yeah, I don't know.
That's okay. Yeah, think on it, or we can talk about it, and maybe I can get to the point there. Any other questions? All right. I want to take a few minutes and I want to go over have any of you started thinking about what your concept note looks like? No. Okay. So real quick, let's see if I can switch over here. Yeah, here we go. I'm gonna have to stop share.
How does it compare to our research proposal work?
Very closely, yes. Right. So all of you are experienced with this. So for those of you that haven't done this, that's why I wanted to go over it, especially for the distance students. But there are 6 criteria here. And when I say high level, again, just keep it high level. We don't have to get in the weeds with it. It has to be related to insect physiology. I'd like to have it, roughly 1 page, which is about 500 words, with relevant citations. I like to limit it to the 1 page because I will get a book from someone and I don't want that. I'm only going to read 1 page. And this is a good lesson to everyone that when you submit grant proposals or anything like that, they have a project narrative or a summary on 1 page and 1 page alone. So you got to get your point across on 1 page, right? So significance, knowledge gaps, and long-term goals. So what I'm looking for here with the significance is that is why the research matters, right? So why is the importance to the field of insect physiology? The knowledge gaps are just what it says, it's just is to what do we currently don't know about that subject, or how your project will address the gap in knowledge, and then the long-term goal, which is the broader overarching outcome of what the project will do. So with innovation, it can be conceptual or experimental. And so when I say conceptual, it's a new way of thinking about something or a problem. If I say experimental, then it could be a new method, right? For being able to provide knowledge to that subject. I also want an approach. So what a general strategy or methods are. Again, you don't have to get in the weeds in this one. I'm just looking for a high level approach. Again, you don't have enough space to write this in depth. What's the central hypothesis, and what is the testable idea that your research will have, and the rationale? Is there a logical reason for doing this type of work? Sometimes we get hung up on just laboratory curiosities, but think about the bigger picture here and the broader impact. Also focus on, what the expected outcome is. And then when I say broader impact, how does your science or what you're working on, how does it contribute to the science, to society, or applications, whether it's pest management, whether it's conservation, whether it's evolution, whatnot. So think about that broader impact on it. And then, of course, I asked for a graphical abstract, which is basically a visual summary. Okay. A lot of you that were in 315, it was 315, right? Yeah, did a really nice job on this. So have fun with it. I will put some examples of past concept notes from the physiology course online so you can kind of see what's happening. They've been all different shapes and sizes before. Some of them have been a half page. Some of them I opened them up to 2 to 3 pages. And again, I'll get someone who writes a 20-page manifesto. We're not going to do that. So any questions?
Should we talk to you about which gaps? It's like, I don't know. I don't have time to read every paper ever written on
No.
mosquitoes.
No, and I don't. Yeah, you can. You can come talk to me about any of your projects if you want to. I don't expect you to be able to fill every knowledge gap out there. I can't even do that. So think about what the topic is and think about what the impact of you working in that topic area. A lot of times that'll lead you into where there's a knowledge gap, right? Sometimes you can just do a simple question search online and it'll take you in a variety of different directions. This is actually where AI becomes a bit useful. Any other questions? Good to go? How many of you have topics so far? Haven't even thought about it, Val.
To do the research I'm doing right now.
There you go.
That was kind of my
I'm going to say all of you, I think most of you have research projects that you're doing. So, I mean,
Yeah, I was.
don't reinvent the wheel. Go ahead and use those for this assignment. What's that?
It's gonna take what? Copepods, they're not insects.
really close.
They are.
I will allow it.
You'll allow it?
Yes,
It's
yes.
an arthropod.
Because I know that you will make it super interesting for Nora, so yes, I'll allow it. Okay. Anyway, I bring this up because they're due in almost 2 months. It's right around the corner. I know that you all have other assignments, and so don't get lost with it. Okay. And I'll bring it up over the semester again. But again, if you're having difficulty thinking of a topic or whatever, I don't think any of you are going to have any challenges with that. Please come see me, and or if you have any questions about what each one of the criteria are, I can do that. Okay. I am done today. Have a good rest of your week. Good
Will,
weekend ahead. Eli.
will Wednesday's class next week be online?
I haven't decided yet. Yeah, I'm supposed to be at University of Florida, but the PI of the student defense has something that just came up. And so I need to figure out. I'm not leaving if he's going somewhere else. So in other words, right? So I'll let you know on Monday. Cool. All right. Again, have a good week ahead and good weekend.